Okay, I'm recording now.
Well, maybe we should wait for him to.
No, it's fine. I mean,
Okay. Alrighty. Thank you so much, everyone, for coming. Thank you so much, Anila, for being here. So, today, as our speaker, we have Anila D'Mello, who's a assistant prof at UT Southwestern, and she did her BA at Georgetown, then a PhD with Catherine Studley at American University, and then did her postdoctoral training at MIT with John Gabrielli. And she's broadly interested in language and cognition and has a special interest in the cerebellar role in these behaviors. And she's also very interested in autism, and we're going to hear about that today. So the kind of techniques she uses in her lab are broadly neuroimaging, and then she's looking a lot into developmental populations as well. So yeah, I'm very excited to have her join today and talk to us about her work on cerebellar-mediated rescue of autism-related behaviors in mice, and also link that to her work in human neuroimaging. So, Anila, take it away.
Great. Yeah, thanks for having me. So the way I kind of laid this out is to give kind of a brief background on some of the work on cerebellum and development, cerebellum and autism, and then kind of dive through and do this journal club style, go through the figures and tell you kind of some of the motivation for why we did what we did. As Carl mentioned, this was a collaboration between a human group and a mouse group. So, I'll try my best to answer any specific questions you have, and we'll see how, I'm a human person, so we'll see how that goes. Okay. So first I kind of want to give you a little bit of an overview. I think this is like preaching to the choir for this group, right? But about cerebellum and cognition, cerebellum and typical development, and then cerebellum and atypical development. So I'll give you like a brief background on that. We'll talk a little bit about the paper and what I think are some insights about the paper, and I'd love to hear from you guys. And then, of course, we can always talk about future directions as well. And so I'll start, again, not really appropriate for this group. I'm sure you guys know this, but with this kind of general understanding about the cerebellum. And so cerebellum means little brain. It literally means little brain. And I think what's important to note is that in the human in particular, it's an extremely folded and foliated structure, much more so than in any real animal, so much more so than in the mouse, for example. And it's a structure that's expanded dramatically over time, right? So like in phylogenetically newer species, it's expanded along with the expansion of the cerebral cortex. And it's so gorgeous that it has inspired some of the famous neuro art that we have in our field, right? So these are the drawings of Ramon y Cajal of Purkinje cells, and these are the main output cells of the cerebellar cortex. And so at the level of analysis I'll be talking about today, the cerebellum is subdivided into these three main lobes, and these are further subdivided into a number of lobules, which are numbered here, with lobule 7 being particularly large and even further subdivided into crus 1, crus 2, and 7b. And all of these regions of the cerebellum are really intricately connected with the cerebral cortex through a series of reciprocal closed loop circuits. So one region of the cerebellum connects to one region of cortex and vice versa. And these are contralateral mainly in nature. And so, of course, what we know, what people still learn today, what I learned in school, what medical students learn is that the cerebellum is a motor structure. It's critical for governing reaching behaviors and coordinating motor behaviors, things like balance, right? And we know that damage to the cerebellum affects these behaviors, right? So it can result in things like ataxia or dysarthria, right? So slowing of speech. And we have a good sense of the fact that there's a nice physiological representation in the cerebellum for these sorts of behaviors. So early physiology showed us that there's actually somatotopic representations of the body in the cerebellum. Unlike in motor cortex, these are represented in triplicate within the cerebellum, right? So you have this upside down guy hanging out, these sideways guys, right, over here. And if you ask people to move body parts in the fMRI scanner, you can actually really nicely recapitulate these kind of somatotopic representations. And you can do this even at the level of single subjects, right? So this is a really robust organization within the cerebellum. And if you ask people to do more complex tasks, for example, articulate words, you actually activate these regions, right? So where the motor articulators for speech live in the cerebellum. And conversely, if you damage those regions of cerebellum, you end up with articulation difficulties and dysarthria. So this is a nice way to say, okay, there's some basis for these motor disruptions that we can see in patients, and you can see this with neuroimaging. But of course, as in everything in life, patients have challenged these canonical ideas about the cerebellum as a motor structure. So damage to the cerebellum can result in a number of non-motor issues, some really weird issues. And when researchers really started to look at what it was that distinguished cognitive from motor symptoms following cerebellar damage, what comes out is the fact that it's the location in the cerebellum that matters. So by and large, if you damage these motor effectors for things like speech or movement, you might get the classical motor syndrome of cerebellar damage. And if you damage posterior regions of the cerebellum, you might get cognitive issues in the absence of any motor issues at all. And in fact, when you look at patients, some of the biggest issues, right, following cerebellar damage tend to be in really high order domains, things like language, executive function, right? So very different than what our kind of canonical ideas about cerebellum are. And so a lot of work by people in this group, right? So Jorn and Caro and others, Basel, others there have really worked on, and our group as well, have worked on kind of reconceptualizing the cerebellum as a structure for cognition as well. And so while, of course, we see these beautiful motor representations, there's a lot of the cerebellum that doesn't seem to at least directly represent motor behaviors, particularly these large posterolateral hemispheres. And so while these motor regions do form, these nice circuits with somatomotor regions of the cerebral cortex, actually the vast majority of the cerebellum seems to be connected both functionally and anatomically to non-motor regions of the cerebral cortex, right? So these are regions that are important for things like language and thought and all of these higher order things that make us human. And this also jives pretty well with evolution, right? What we know from evolution. So it turns out that the cerebellum has expanded over time, just like the cerebral cortex in phylogenetically newer species. But in particular, these large posterolateral hemispheres have expanded almost linearly with the expansion of regions like the prefrontal cortex, and sometimes even outpacing the expansion of prefrontal cortex, or at least what would be expected for someone of our human size. Okay, and so just 2 more slides on this, but I think it's interesting to conceptualize cognitive functions in the cerebellum similarly then, in some sense, to motor functions. So just like there's this topography of the body, right, in the cerebellum, you also have a beautiful functional topography for cognition in the cerebellum, whereby different types of functions are located in different parts of the cerebellum. And the location of these functions in the cerebellum is kind of expected based on what we know from their location in cerebral cortex. So for example, language is largely right lateralized in the cerebellum, and that's consistent with its left lateralization in cerebral cortex. Okay, and so my lab has kind of, and my work in grad school and postdoc has kind of tried to say like, okay, well, how far can we take this concept of a cognitive cerebellum? And it turns out that in things like sentence completion, where you have to understand, right, the semantics of a sentence, when you're judging whether a sentence, not whether a sentence is correct or incorrect, but whether it's plausible, whether it could happen, or implausible, you see activation in the cerebellum that is as high as activation in the cerebral cortex, in regions primarily of the cerebellum that we'd expect based on what we know about this functional topography. We find that the cerebellum is really engaged in social learning paradigms, and we've even described these really gorgeous kind of hierarchies in cerebellum that kind of mirror what we see in dorsolateral prefrontal cortex for things like different aspects of cognitive control. And relevant to this paper that we'll talk about today, we've done some work with neuromodulation of different cerebellar regions, showing that maybe there's some specificity in how we can modulate cerebellum and what kinds of networks or behaviors we can affect long term. And so, just to kind of really hammer this, beat a dead horse, I guess, right? It seems like the functions, right, these really higher order hand wavy almost functions that we ascribe to cerebral cortex are also very much being represented in cerebellum, right? So this is a study that we just published where we looked at the content of language, right? So like how interesting were these stories to different kids that they heard in the scanner They were personalized to individuals, right? So they were maximally interesting to the person. And, we also see alongside these kind of canonical language regions in the cerebral cortex that the cerebellum is also doing this kind of, interested in the content of these stories themselves. Okay. So I think you're convinced, and I probably didn't even have to go through that, right? That the cerebellum is doing something for cognition, and some of these really, really high order aspects of cognition that are going to become important in disorders like autism, which are really conditions that primarily affect these kinds of higher order functions. So something particularly interesting about the cerebellum is that we think, and many others think, that it may actually play a really key role in development. Right. So this is, my daughter's, she has a cerebellum. Great. This was when she was 18 weeks old. This is the baby I'm carrying now, about 5 weeks ago. They have a cerebellum. Looks great. Got to make your stenographer check during your ultrasound whether your baby has a cerebellum. They do. Great. And so why, right? So why is it that the cerebellum is so important in development? And so I want to kind of tell you this little story, right? This tale of two brains. To kind of elucidate this point a little bit. So this is a study of a woman who was missing her entire left temporal lobe from birth. And this is a story of a woman who didn't realize until late in life that she was missing her entire cerebellum. So this woman over here was fine, right? She completed college. She had a great vocabulary. She learned a foreign language. And she lived life pretty well, especially language, despite the fact that she's missing kind of this critical cortical hub for language, right? But the story is actually quite different with this woman over here, right? So this is a woman who didn't stand for the age of 4, didn't walk until the age of 7, never ran, never jumped, and had such intelligible speech past 6 years of age that she didn't ever enter school. Right. So she wasn't actually able to get an education. Now, in this paper, it's in Science Alerts, this is described as minor ailments. They seem pretty major to me, right? But she's pretty impaired, right? Having lost this part of the brain that we don't typically associate with cognition or stuff at all. Okay. And so what's interesting about the cerebellum then is it kind of seems like it's doing something different than the cerebral cortex. So in the cerebral cortex, it looks like early damage might allow for plasticity and repair. And this is canonically what we learn about in school, right? This is a period of maximum plasticity. So you have early damage that leads to better outcomes. And if you have damage late in life, you actually have a lot of issues potentially. But in the cerebellum, we see something slightly different, and that is that earlier damage to the cerebellum seems to result in worse long-term outcomes, and worse long-term outcomes that are permanent across the lifespan. And there's some reasons I'm happy to talk about why this might be the case. Okay. All right, and so that brings us to disorders, right? So if the cerebellum does all this stuff for language and cognition and social communication, if early cerebellar damage might, be worse than cerebellar damage later in life, might there be some link between this structure and neurodevelopmental disorders? And so the disorder that we study in our lab is autism. And so for those of you that don't know, autism is a neurodevelopmental condition. It currently is estimated to affect 1 in 36 kids in the US. And although it's primarily characterized by social communication difficulties and the presence of repetitive behaviors, it's actually, a spectrum, right? So there's a lot of different kinds of aspects of autism that any individual with autism might deal with: executive function issues, sensory issues, et cetera, et cetera. And so then the question becomes, okay, is the cerebellum relevant in autism? And what's interesting is that actually across animal models, postmortem studies, neuroimaging, it turns out that the cerebellum is one of the most consistent sites of differences. So we know from early studies that tumor resections in the cerebellum can result in pretty extreme autism-like symptoms. So this was a girl who had a tumor resection in her posterior cerebellum, and her behavior changed in this startling way. So she started rocking stereotypically, rhythmically, moving her hands, kind of like these repetitive behaviors we see in autism. Spontaneous language disappeared. She had echolalia and actually met diagnostic criteria for autism.
Is that patient, how old was she when she got the result?
I think she might have been like 6 or so. I have to go back to the original paper and check, or maybe younger, 5 or so, 5 or 6. So she was not a baby. She was a child. Yeah. She started cursing, so that's not great for a child. And so, right, so this is some of this evidence. And actually, when you start to quantify cases like this, it looks like something like cerebellar injury at birth is associated actually with a higher probability of getting an autism diagnosis later in life than even having an autistic fraternal twin, right? So this is like a 36% increase or times probability increase, right, of receiving an autism diagnosis. And this isn't new. So in the 90s and early 2000s, Eric Courchain primarily, but many others published a lot about these findings, right? They found cerebellar differences in autism, and they were kind of all over the place. And in these early works, so this is published in 1997, right? They found that pathology in autism, so for example, reduced number of Purkinje cells, were primarily located in areas like the vermis, the posterior vermis, and also the posterior hemispheres, right? And so this is, so Eric Courchain at the time really focused on the size of the vermis, and this is just showing you, a lot of different studies in the early 90s that found kind of these size differences, right, using MRI in the size of this cerebellar structure. And then, it kind of fell out of favor. So people just kind of forgot about it, or it wasn't, followed up on too much after this. And so in some of my graduate work, I was interested in following up on some of this work. And so we, with kind of renewed methods, right, renewed imaging methods, kind of took a look at this again, right? And so this is just one study showing that in a group of autistic children, when we start to look just broadly, these are group differences in gray matter. One of the largest areas that comes out as being different is this right posterolateral or posterior, let's say, cerebellar area in CRUISE 1 and CRUISE 2. And actually, when we do some correlations with behavior, it looks like all of these core behaviors in autism, all of these core symptoms, seem to correlate with gray matter in this region. So if you have worse social interaction or worse repetitive behaviors, and you're autistic, you have reduced gray matter. And that kind of converges on this region of CRUISE 1 and CRUISE 2. We also looked at the size of lobules. So we took like a multimodal approach to do this. So not only looking at individual voxels, but also the size of various cerebellar structures, and find kind of similar things as some of the early Courchain work. And we also looked at kind of subtypes of autism. So in early days, right, before the DSM-5 came out, you could have a diagnosis of Asperger's, or you could have a diagnosis of autism. And really what differentiated those two was the presence or absence of an early language delay and things like intellectual disability. And so we looked at whether early language delay also looked different in the cerebellum, right? And so once again, we found that, yes, it looked like individuals with early language delay. Actually, the cerebellum was the only region we found that really differentiated these groups. And it looked like individuals with early language delay had this kind of double hit, so this bilateral hit in CRUISE 1 and CRUISE 2 that was associated with their language delay. And in fact, the degree of gray matter reduction in some of these posterolateral regions was associated with their language onset. So more gray matter reduction was associated with the later age of language onset. Okay, so yeah.
I was wondering what's happening in the cortex for these studies. Like, do you see any size and gray matter reduction in cortex as well?
Mm-hmm. So in this study, we did see some cortical stuff. It was robust, but not as robust as the cerebellar stuff. In fact, and in strange regions, which end up coming up a lot actually in autism work, including visual areas, which was interesting. In this study, we saw nothing that differentiated these groups. at least in terms of these just group differences in cortex. When you start looking at correlations, the direction of the correlation, and I'll talk about this a little later, does show you some cortical stuff in parietal areas. But when you do these kind of group analyses, yeah, it was just cerebellum, which felt almost fake to us, right? When we first did the study, we were like, what? How could this be? But it was the most robust difference that we could find.
Thank you.
I was also wondering, just since you're showing this right now, so the fact that you have in Asperger's then still the social impairments, right? But you get this left cruise-one difference, right? To I assume like neurotypical.
Yeah. So basically, it's either like a bilateral effect or a unilateral effect in just like non-early language delay. And it wasn't, an exact Asperger's versus autism comparison, right? So it was really, we sorted only on language delay. So did you have a language delay? Did you not have a language delay? It turns out that that is the kind of deciding criteria, or it used to be before Asperger's disappeared, right? So you can't have that diagnosis anymore. But yeah, so we kind of thought, so yes, the left crus one ends up being the difference between the groups, right? It comes out as kind of differentiating the two groups. But when you look at both of them versus typical kids, it's really that one has this bilateral deficit and one looks more unilateral.
Anila, I don't want to go down a rabbit hole, but how does the DSM-5 now carve up autism?
So DSM-5 got rid of, so basically they said, early language delay versus non-language delay, it doesn't actually end up being different in terms of outcomes, which could be argued, right? And so they got rid of that Asperger's diagnosis, and everyone has this autism spectrum, right? And so the criteria in terms of meeting the diagnosis are the same, right? You have to have basically the two main ones: presence of social communication deficit, presence of repetitive behaviors, so more or less the same. And within those, right, you have to meet a checklist of having, let's say, at least 4 examples from each of these pillars. And they can include things like sensory problems, right, for in the repetitive motory domain, or, hypersensitivities, yeah.
Okay, so they have a big bucket now, diagnostic.
They what?
They use a bigger bucket now.
They use a bigger bucket, but some people felt at the time that some people would lose their diagnoses, right? These people kind of on the border. It kind of looks to me like, if anything, now the diagnostic criteria is like inching up to almost typical, right? In terms of who's getting diagnosed and in terms of the numbers that are getting diagnosed. But yeah, at the time, people were nervous that this would kind of cut some of the low support needs or higher functioning individuals out. Yeah.
Sorry, just to finish the point earlier. So were you surprised to see that the difference between the language impaired versus or language delayed versus non-language delayed was actually on the left rather than the right in the slide?
Not surprised, because the way we interpreted it was the fact that one was a bilateral. So basically, like, the way we interpreted it, right, was that if you have autism, you have this right Creux-Swan difference. If you additionally have a language delay, additional regions of the cerebellum come online. So the difference being in left Creux-Swan, I think, was not as much of like, oh, this is what's differentiating the groups, as the fact that it's like this unilateral difference versus like even more of the cerebellum being involved if you have an early language delay. You know what I mean?
Okay, yeah, I think, yeah.
Yeah. We were surprised that the cerebellum turned out to be as robust, because it's kind of like, feels almost like cheating. Your cerebellar group, you find a cerebellar difference. Seems almost too good to be true. But right, this is what the data showed. Okay, so when I was a grad student, I used to tell my mentor, Catherine Studley, all the time, I think right Creux-Swan is for real. Like, it's for real, it's for real. We kept finding it right in autism. And in fact, even in meta-analyses that were done by Catherine Studley, so she was my graduate mentor at the time, you do kind of consistently see this right Creux-Swan region. And this actually kind of makes sense with what we know, right, about about the way the cerebellum is organized. So this is a region that's really important for things like language, for higher order skills like, narrative processing, social cognition that both Caro and, Maeve have identified as well. Okay. And it actually jives really well with what we know from the animal literature. So in mouse models of autism, it turns out that cerebellum is also pretty consistently different, right? So this was a study by Peter Tsai showing that in this tuberous sclerosis mutant mouse, you have atypical morphology and density of Purkinje cells and also a number of social issues. So we explored this in this paper. So, okay, just checking out the time. We explored this in this paper where we thought, okay, we have all these cool tools. We have the ability to really target a circuit really well if we want to in the animal. And so let's look at, what it is about Wright-Kruse-1 that might be so important for autism. And so we can talk about the paper now. I'll tell you a plug. We made the cover of this Nature Neuroscience. I drew this cover. My parents were so proud at the time that they made shirts. So we have matching shirts now of this cover. And so so I'm happy to kind of step through. Does anyone have any questions before we kind of like step through, the paper just briefly?
I think so. Let's go.
Okay. So the idea, right, of this paper is to kind of really characterize what it is that's important about Wright-Kruse-1 and what it is that it's doing at the circuit level and for behavior in autism. And so, the first thing we did was really just kind of recapitulate what's already known. That Wright-Kruse-1 is connected broadly, both the whole lobule, which is really not the way to do this, but individual places within this lobule with regions of the cerebral cortex that we think are really important for language and thought and, all of these higher order things that are also implicated in autism, right? And so we think that targeting Wright-Kruse-1 should affect regions and networks that are implicated in these behaviors in autism. And so in this study, we had 33 adults that, completed this imaging paradigm, and they also completed some sort of neuromodulation. And the neuromodulation that we chose was something called TDCS, or transcranial direct current stimulation. This is really a pretty like low-level technique, right? So it's essentially a battery that's connected to two electrodes, and these electrodes are placed on the scalp. And depending on the direction that you place the electrodes, you can get current running, basically. So basically, current runs between these two electrodes, right? An anode and a cathode. And so depending on the electrode placement, you can get different polarities of effects, either hyperpolarizing or depolarizing, regions under the site of the electrode. And so we did this within the MR environment, and the reason being that the effects of this are pretty transient, right? So it's like a 45-minute effect. And so we wanted to kind of have people in the scanner while they were getting TDCS and then look pre-post at how their networks changed. Okay, and so what we did was we applied anodal TDCS to Wright-Kruse-1 of the cerebellum. There was nothing fancy here, so we defined this based on skull landmarks, like the way you would EEG or something like that, right? And what we found is that when we did this neuromodulation of Wright-Kruse-1, we were able to alter whole brain circuits. And the region that we found the largest difference in was this region in the parietal lobe. So this, this IPL region here. And I don't usually show this because I don't know that, it's relevant, but I think something striking is, sometimes it's hard to believe that like such a rough technique actually has an effect, but actually, this is every participant in each of the groups. So this is the sham group, this is the anodal group, and it actually looks like right on the individual subject level, the directions, right, of these effects are consistent, right? So it looks like in almost every anodal subject, things are moving in one way, and in every sham subject, things are moving in another way.
You get reduced correlation in resting state after the application of TDCS.
Yes. Yeah. So this becomes a big sticking point, right? About like what does functional connectivity mean, right? What does the correlation coefficient mean? And whether it's good or bad. versus versus what's happening kind of like at baseline.
This is like 20 minutes after TCS application or something.
This is 20 minutes of TDCS application, right at like 1.5 milliamps or 2 milliamps, and this is right after.
Right. Mhm.
And they were doing nothing during the. They weren't doing anything, right?
They're not doing anything. Yeah, it's a resting state. Yep.
Yes. The rest.
Okay. So then so that's what's happening in the human. Okay. Now we can do the same thing, except not with TDCS and little electrodes and little batteries, but similar, right, in the mouse. So here, instead of TDCS, yeah.
Do you know anything about the spatial resolution that you were like? Do you have any way to estimate how precise you were stimulating that specific part of the cerebellum?
With the TDCS?
Yeah.
Yeah, so no, right? So we've done modeling and things like that, or I've kind of moved a little bit away from TDCS, but the modeling shows like you're relatively getting right these posterior regions, but it moves much beyond the posterior regions. I think we're lucky in the sense that like the region we want to target happens to be the most massive region, and that it's like kind of located perfectly, maybe closest to the scalp. What we did in this paper was like we looked at functional connectivity changes from the lobules or the regions right adjacent to what we thought we were stimulating and determined whether functional connectivity changed from those lobules. And so we didn't get any functional connectivity changes from those. But
you didn't test changes in functional connectivity in other parts of the cerebellum, of the same hemisphere that you were stimulating.
No, they did.
Oh, they did. Okay.
Yeah. So adjacent lobules.
Oh, okay.
And there were no changes, right? Yeah.
no changes.
But
Yeah.
Directly under the electrode there were.
Right. But that could also just be like lucky for us in terms of like what we are stimulating. Like if you were trying to stimulate something different, that might not have been the outcome, right? So like that's not necessarily speaking to how great the technique is. It's more speaking to like the fact that the lobule we were interested in modulating might just be so big that the fact that effects were spread was okay for us. Do you know what I'm saying?
Yeah. Thank you so much.
So the cathode was where in this case?
The electrode?
Yeah, the cathode. The anode was right.
Oh, on the like right here, like on the clavicle.
So
On the optic.
I see.
That was the montage. Okay, so that's our human neuromodulation setup. So now at the same time, we can do this in mice, and we do it a little bit differently. So this is in collaboration with Peter Tsai, who's a pediatric neurologist here at UT Southwestern. And so what he used to modulate the cerebellum was chemogenetic inhibition and excitation using something called DREADS. So for those of you that might not be familiar, these are designer receptors exclusively activated by designer drugs. And so the kind of basic way this happens is that you can selectively inhibit or enhance responses in particular cell types with temporal specificity as well, right? So in our case, we were interested in Purkinje neurons of the cerebellum. So you have a group of mice that's expressing this CRE in Purkinje cells. You can then via injection, right, via a viral vector, deliver DREADS to right CRUS1 specifically, right? These CRE recombinase dependent DREADS. In then only CRE expressing neurons, you can remove, so this is like a CRE LOX-P system, right? So you can remove the stop sequence, allowing these DREADS to be expressed and whatever the inhibitory DREAD is to be transcribed. So in this case, this is an inhibitory DREAD. They also did this for excitatory DREADs in mouse models of autism. And then upon administration of a certain drug like CNO, that can activate or inhibit these specific neurons, right? So it's a similar setup in the sense that you are, well, not similar in the kind of specificity, right? But that you're specifically inhibiting or exciting Purkinje neurons. And because of the DREAD injection, we can say that we're doing this in right CRUS1 versus left CRUS1, right? Okay. All right, so when we do this broadly, right, what we see is that with CNO, you get reduced firing frequency in Purkinje cells. You see this too. This is in slice recordings. You can do this in vivo as well. So this is showing right CRUS1, the sites of the injections, right? And this is also in vivo. Then you can see that upon, and this might be in the parietal lobe actually. Yeah. So then upon CNO administration, you get not only a reduction of firing in right CRUS1, but when we look at parietal regions that are defined based on the parietal regions we find in humans, in mouse, so they're mouse homologs, we find increased firing in these regions. Okay, so we're kind of now mapping a similar circuit, let's say, right? So we're reducing, in the mouse, we're reducing, inhibiting firing in Purkinje cells in right CRUS1, and we see this concomitant increase in firing frequency in this parietal region, just like kind of you might see in the human, right? Okay, so the basic point here is that neuromodulation of this specific region in the cerebellum looks like it's affecting a similar circuit in mouse and man. And the question then becomes like, why should we care? And is this relevant to autism? And so what we did, so this is in collaboration now with Jason. So this is the same paper, but in collaboration now with Jason Lurch, who's actually at Sick Kids up in Canada. They did some mouse imaging first of a autism mouse model. So this is the TSC1 mutant mouse that shows autism-like symptoms when TSC is affected in Purkinje cells. And so in these in these mice, right, you also see structural differences in parts of the brain that we're particularly interested in. So right now, we're interested particularly in these parietal regions that we've identified in our humans as being different. Or being affected by neuromodulations, and you see similar structural differences in these regions. And actually, when you quantify, for example, the correlations, let's say these are structural correlations between the structure of CRUS1 and the structure of these lateral or medial parietal regions, you see that in the control, right, you get this negative correlation, and in the mutant, something shifts, right? So reduced CRUS1 is associated with increased volume in the medial parietal lobe.
But this is very bilateral, right? This is not necessarily the right side.
Right. So this was, it was, I think, stronger in right CRUS1, right? But quite bilateral. Yeah. Okay. So now we can say, all right, well, this circuit, we can affect it with neuromodulation. The circuit looks strange in the mutant mouse. What about in autistic kids or autistic adults, right? And so for this, we got a huge amount of data from Stuart Mostovsky's group at Johns Hopkins. And they looked at a really large sample of autistic kids and neurotypical kids. Using the same seeds that we had used, right? So the seed in right CRUS1, this left parietal region that we had identified. And they too find, now this is, again, this is functional connectivity, right? What was happening in the mice was structural connectivity. But they also find this kind of atypical relation, right? So that they find increased functional connectivity in this circuit. Okay, so kind of mirroring what you see in the mouse.
The seed includes 1 year. Is this more in a, would you guess that it's more in a multi-demand region? So kind of like executive function, or is it more like in the social linguistic circuits?
I don't know, like, this was such a long time ago that I don't know that I have mapped it really well. It's kind of like a strange area. So I don't know. I need to go back and look at like a map of exactly where this IPL cluster is. Now, right, in retrospect, we do find, right, in that same paper where we found early language delays, that if you flip the correlation, so if you look at, so we looked at regions where decreased gray matter was associated with later age of language onset. But if you flip the correlation and say, well, okay, are there regions where there's increased gray matter that is associated with age of language onset? We find the same region, right? So that suggests the same IPL region, right? So that suggests that maybe it's associated, this region basically is associated with a lot of things, right? So if you put this in like neurosynth or whatever, you get like everything, like tool use, you get, you know, imitation and praxis, which is like a big issue in autism. Yeah, so unclear, relatively non-specific. But I haven't looked exactly at like where it falls into like network maps. Or I didn't at the time. Okay, so these directional effects, right? They look like they're all over the place, but they actually end up aligning really well with the mouse data. Okay, and so now something we can do in mice that we can't do in humans, right, is to actually modulate right crus1 and see what happens to behavior. And so if you inhibit right crus1, you and look at things like social approach behavior or social novelty behavior, right? So how much time does a mouse spend with a novel animal versus a novel object, for example? What you can see is that typically wild type mice spend a lot more time with a novel animal, right, over a novel object. If you inhibit right crus1 specifically, you can kind of obliterate that normal interaction. This isn't the case, obviously, under any sort of vehicle, right? This is specific to this CNO manipulation. And it's also doesn't affect left crus1, right? So you could do this in left crus1 and you don't see any kind of effects on social behavior. Same with things like social novelty.
Yeah, and has anything like vocalization or any kind of language very specific functions? Just because
No, I don't think we did vocalizations. I don't think we did vocalizations. We tested a number of other things. So beyond social novelty and kind of like grooming or behavioral inflexibility, where we also see these differences. So you can see down here the grooming differences. We tested a number of like controls, or Peter's group tested a number of controls, right? So like visual issues, locomotor issues, olfactory issues, and they didn't really show any differences there, right? So, so that's
here. I'm just trying to wrap my mind around the fact that this is all fairly specific to right crus1.
It's really specific.
What we usually see, or what we've seen lately in the data, is that it's really kind of the left side that's more responsive to some of the theory of mind and emotional recognition. And I think Dagmar's group in Germany also found that it's like emotional recognition, at least fear conditioning, which is, you know, not the same, but still emotional kind of stuff really sits more on the left than on the right.
Right.
So I'm wondering whether there's
which makes sense, makes sense, right? Based on kind of some of the social stuff, like, right, no, if you're like into the right TPJ as being right important for social behaviors and stuff like that. So yeah, so that would make sense to me, right? That like maybe social cognition is like more right lateralized, more left lateralized in cerebellum, given that it could be more right lateralized in cortex. Yeah, right crus1 ends up, I mean, so it's not like we never see left, right? But it ends up being, if you look across the autism literature, like really consistent.
Yeah.
And yeah, the question now becomes why. From a language perspective, I think it makes a lot of sense.
Yeah.
From a social perspective, it's unclear, right? It's unclear what the difference might be. So if you do like TOM type stuff, you get both left and right pretty robustly, right?
So, sorry, I have a quick question. At what developmental stage were these mice when you did the manipulation? Meaning that, did they ever learn how to do like a normal social interaction before?
Yeah, so this is, I think they get the dread at like 5 to 7 weeks. So pretty, not like late, but they're not like infants, right? And then I think they get the CNO, because the dread takes a few weeks. They get the CNO at like 7 to 9-ish weeks, right before they do the behavioral manipulation. So young adult, like adolescent to young adult, right? So these aren't like teeny tiny, like useless. They've never had a social interaction before.
Do you think that we would see the same result if it's done in like completely adult mice?
I don't know. Yeah, that's a really good question, right? So like whether there's, and in the paper, we do talk a little bit about like critical periods and like whether, and you'll see, you know, in the rescue that some of these behaviors are rescuable and some aren't. And that could also have to do with developmental stage, right? And the fact that maybe there are critical periods for some of these things and not other things. So yeah, that's a really good question. In my mind, the fact that you see it in, let's say, adolescence or early adulthood is really promising, because here we're not talking about like a preventative measure. We're talking about like a treatment type measure. And so, that's interesting, especially if we think that like learning social behaviors does have some sort of critical period in early adolescence or early childhood. Maybe it does, maybe it doesn't. So yeah. Yeah, okay, so we see these social issues. I circled here, we also see these kind of repetitive behavioral inflexibility issues. So this is really recapitulating the phenotype, let's say, right, of autism. As Cara mentioned, this isn't a result of locomotor issues, licking differences, visual issues, olfactory issues, at least not that we could find. And so the summary kind of at this point is that, okay, both the mouse model and kids show atypical Wright-Kruse-1 structure. Both the mouse model and autistic kids show atypical connections with this contralateral IPL, and inhibition of Wright-Kruse-1 results in ASD-relevant behavior and increased firing in IPL, right? Okay, so then the question becomes like, what do we do, right? Can we do anything? And so now what you can do, again, in mouse and non-human is you can excite Wright-Kruse-1, you get, the opposite effect, right? So increased firing, decreased firing in IPL, and rescue this ASD phenotype, right? So now if you do social approach, so I should mention, these are the TSC1 mutant mice, right? So, and with TSC expressed primarily in Purkinje cells specifically. And so this the mutant mouse shows no difference, no preference for the novel animal over the novel object, right? But then when you administer, right, CNO, so when you're exciting Wright-Kruse-1, you all of a sudden see this kind of rescue of behavior where you recapitulate the wild type behavior, right? So now they're interested in the novel animal, they're not interested as much in the novel object. You can look at time in chamber, you can look at time sniffing. What's interesting is that you don't get a rescue of every behavior, right? So you get a rescue of this social behavior, you don't get a rescue of this kind of grooming differences, behavioral inflexibility differences. And so the thought is, like someone mentioned, maybe there are critical periods that kind of close for rescue of some of these behaviors. Okay, so just a really quick summary of the findings. If we inhibit Wright-Kruse-1 and look at behavior, we change this behavior, we do the opposite, we rescue this behavior. We think that one of the circuits is this kind of Kruse-1 IPL circuit, and they act in this opposing manner, right? So you decrease firing in Wright-Kruse-1, and it results in this increase in firing in the IPL. This is similar kind of to what we see structurally, where decreased gray matter in Wright-Kruse-1 in the mutant mouse is associated with increased volume. I won't say gray matter, I'll say volume in the IPL. Similar kind of story in kids, right? Decreased gray matter in Wright-Kruse-1, increased volume in IPL, this, kind of converse circuit functional connectivity. And then of course, our finding in adult where we can decrease this kind of atypical cerebelloparietal connectivity potentially in this group. And that's kind of it. So, and I'm happy to talk more about like future stuff, but I want to maybe give us time to also talk about the paper or anything if people have questions or whatever.
Oh, I have almost just like, how about the opposite? If you modulate the left intraparietal region, then can you modulate the effect? I mean, similarly, like when, yeah.
Yeah, I have no clue. We haven't done it. That's a good question. Yeah, it'd be interesting to think about, like, could you modulate any part of the circuit, the network, and get a similar effect? That would be nice, right? Yeah.
I was wondering, so you also, I read in there that you also tried stimulating on the left, right? And there were no rescue effect, like you couldn't rescue those same social behaviors. Just to understand correctly, was the left also reduced in Purkinje cell firing, or was it just in these mice functioning as normal?
No, because the dread was only injected. So yeah, yes. So like, no, because we were specifically trying to target Wright-Kruse-1, right?
Right.
Yeah.
So okay. So that means they could have essentially had normal functioning on the left. So the stimulation just, you stimulate it, normal functioning doesn't.
Right. Or when you inject right into the left, right, and you do reduce firing there, it doesn't have the same effect.
Yeah. So that was, yeah.
Yeah. Yeah.
Interesting.
Yeah.
Question. We're moving the laptop. So I was wondering, how do you define language onset? So what are the characteristics of that cognitive ability?
Yeah, so in that study, it's a parent report measure, right? So that's not ideal, but it's a parent report measure and it's a standard kind of measure. And you can define it in a number of ways. You can define it as first the age of first word. And so this is something that like if you have a kid, you go to the doctor, like you're asked, multiple times about. And then it'll also ask about age of first phrase, right? So there's a word onset measure, and then there's a phrase onset measure where they're, now speaking a 2-word sentence or more. And so those should occur kind of at different times, right? But the onset of the first word should be close to a year with some, obviously, variability. And so I think the definition for like a delay, I can't remember exactly, but I think it's like a first word after maybe 24 months or maybe 18 months or something like that. And a delay of first phrase would be like after 36 months. So there's standard kind of definitions for what is a delay.
Is it assumed that the language onset is related to the social cognition?
It's not necessarily assumed, right? But it is true that like language tends to be one of the best predictors of outcomes in autism. So kids who learn who acquire language later, who start speaking later, tend to have worse outcomes. So in many domains, right? Now it's not clear exactly why. Like, is language causal here in terms of then the development of social stuff, or is there some other mechanism that keeps language from developing on time to begin with? And so that's unclear. But yeah, language is one of the most predictive measures, and it's actually one of the first reasons that parents seek out evaluations of their kids, right? It doesn't tend to be like sensory issues or this or that. It tends to be language delays.
Thank you.
I have a question if that's okay. Thanks for the talk. It was really great. I think at this point, I'm convinced that cruise one is associated with social
It's for real.
Yeah, it's for real. But one thing that I'm not sure yet is whether cruise one is supporting these sorts of behavior through the computation that should be done always, or whether this is a learning issue. So I was wondering if you can talk a little about that, or maybe if there is a stroke study specific to that cruise one area. So just can give us more information.
Yeah, I mean, I think it's hard to parse apart the computation from like the learning aspect, especially because the deficits are so much worse in kids. I mean, Jordan could probably speak to this as well, but yeah, there's plenty of stroke studies, malformation studies, whatever, that find that these cruise one differences are specifically related to kind of like these like sociolinguistic issues. It's hard to know, right? It's hard to know if like you're disrupting. Are you asking? Yeah.
Future study with the lesions, the tumor resection. Well, we're going to talk about that because that might address.
Yeah. So that is a question, right? So a study that we're doing now, right, is we're recruiting adults who had very early tumor resections in these kind of critical periods for language learning, for example, right? So let's say between 0 and 3 or 0 and 5. And we're doing this kind of precision fMRI mapping, right? So we're trying to say, okay, like what exactly is affected in each individual? We do a huge behavioral battery to really characterize. A problem with some of this is the characterization of some of these populations. It's just not great, right? You do like a vocabulary measure, and that's their measure of language, and that's not really good enough, right? So we behaviorally characterize them. And the ultimate goal, right, would be to say like, well, what is a pattern that supports better language outcomes or better cognitive outcomes? What is a pattern that doesn't? There's so much heterogeneity, right, in the population. So that's something we're doing right now. So like we've recruited like 3 or 4 patients, but it's slow going, right? It's really slow going. But it does kind of approximate like maybe what you can do in the animal, right? This idea that you can like remove the effect of a region and the system would develop more or less without that region for a certain amount of time. And of course, then you could deal with, you could examine the effects of time. So what's the difference between 0? Someone asked, like someone who's never interacted socially, what's the difference with 5? Are there interactions with the type of behaviors, right? Like is language worse if you affect it earlier versus later versus something like reading, right? Which emerges at age 6. So there's a lot that you can do there. And yeah, with regard to the computation, it's hard to know what the computation is and if the computation is learning, and then that's all sorts of confounded. So
Thank you. So we just have to wait a little bit, I guess.
we just have to what?
Just have to wait for the results to come out, I guess.
You'll just have to wait. Yes, you'll just have to wait for the results.
Right. I think that was probably all the questions. Thank you so much for this really, really nice in-depth overview. I guess. Thank you. All right.
And congratulations.
Oh yeah,
Thank
congratulations. That's right. That's right.
you.
Thank you.
Thank you. Bye bye.
Oh, bye.